Good evening, everyone. It's good afternoon here in lovely Australia. Yes, I'm going to talk to you about genetics, and just hopefully by the end of this, I'm going to show you how easy it is to screen. It can be quite painful and difficult, and the key words you're going to hear from me today is personalised medicine, and how personalised medicines and life plans are changing the way you manage your clients in that pet area. So I thought, you know, a lot of people get a headache, but I'm going to assure you that by the end of this webinar, all you're going to need is a packet of throat lozenges to help you clear your throat and understand genetics. Here I was thinking that everyone's going to be in the dark over this, but by the end of my talk, I'll hopefully share the light with you and you'll make it even easier. So hopefully I'll make it even more simple than that for those that have used it. So, but it can be a difficult pill to swallow, I like to call it. And, you know, using genetics and, you know, getting sort of started can be confusing. And usually the confusion is, you know, where do I start? Where do I, you know, maybe source information? Some also find it quite expensive. So the buy-in is quite expensive. You know, do a single test, and some organisations can copy up to $90. So I'm going to sort of show you some of the ways you can tackle that. And I also hear a lot of messages where some vets say, you know, it's not relevant. It sort of doesn't tell me whether an animal's actually showing those symptoms, but, you know, and I'll explain all those. So hopefully by the end of this webinar, I'll be able to show you places where you can source current relevant tests. I can show you just the technology and how it's assisting us to make testing less expensive. And it's really, the information you get is actually quite relevant. So I'm going to talk a bit about clients, because clients are important in the way you manage genetic testing. Because by understanding your client, I think you can understand what type of genetics, you know, who is your client base? Are they a pet owner? Are they a breeder? Are the tests relevant? What sort of test does a breeder versus a pet owner wants? And I'm going to keep on using this word of personalised genetics, and that's the target market we're looking at. And with that comes the pet parents. And I'm going to show how we create this sort of life plan, which builds your client's base, and you're able to develop a schedule for that client. So it's really about where you want to take pet care for your client. That's the key message. So I'm going to talk a bit about clients, because I think by understanding the client, you're able to sort of work out what sort of testing you need. And these are some of the questions that I always get about why a pet owner might select a vet. A lot of them think it's price. A lot of them it's the hours that they're opening. A lot of them and how they treat, how the vet treats the owner and their pet. But most of them sort of say it's cost, but I don't think it really is cost. If you personalise the experience for the client, I don't think cost is a factor. And I like to sort of break it down into the key message you're getting across. Is it that the client says, I went to my vet and they told me about my dog's whole lifestyle? That's the message you need to be getting across, versus, you know, the way vets used to do medicine, which is, I read on the internet that a vet gave a F5 vaccination and caused cardiomyopathy. So cost isn't a factor if you're able to personalise the experience for the client. I also like to sort of talk about some of the things that, you know, can pretty much peeve you off about some clients. You know, clients can be difficult. I deal with them. I deal with them on a daily basis here, and I'm still trying to sort of develop a business where I can make money and not have clients. I don't think one exists, to be honest. So we have to deal with them, and we have to work with them. And these are some of the peeves as published by Christina Lotz in the January edition of a vet journal. And most of you hate it when a client comes in and says, oh, I read that on the internet. I get the same thing here. Oh, I read this on the internet. You must be able to do it. You know, if you're able to sort of offer them some scientific information and clarify what they've read. Sometimes we hate when they wait too long before they come to see you. You know, they won't bring the pet in until it's almost dead. You know, classic quote, he's been vomiting only 2 or 3 times a day. So There's that cost factor where some are reluctant to pay for vet care. Some of them sort of taking that responsibility. Some of them are not sure, you know, whether it's suitable for their environment. Handling the owner, how to handle the dog, you know, pet parents, they're becoming more and more a parent now. It's not like it was years ago. They now like to take their dog with them when they have a coffee, socialize with them. It's like letting go of their baby. And the last one, which everyone hates, is when they use their mobile phone. But I do think if you understand your client, you can manage it and manage what they need. And I always like to finish with this sort of quote from a veterinarian, Dr. Pedro Mancini from São Paulo in Brazil. He was an ophthalmologist. When he was posed with the question, if there was one thing you could imagine that would improve the veterinary profession, what is it? And he sums it up fairly well in that it's the people skills. You know, you tell your students and your interns that, you know, much like you have to learn to deal with owners, and you get so many variabilities in these owners. The different way they talk, their worries, their laughs, their fights. So I just wanted to give you a feel that, you know, all this comes down to being able to manage your client. So let's look at the way testing is expanding and our patient-initiated testing. You know, we all, some people now wear a smartwatch. You know, some other people have, you know, monitors to monitor their health. The same thing is happening in the pet industry. We're seeing it. We're seeing how technology has increased by 37%. People are taking it up by 37%. Things such as collars to monitor health. You know, there's plenty of tracking devices. There's even, you know, food bowls that you can buy to train your pets. So people are starting to take control of the way they manage their animals, just the way they're taking control of managing their own health. So it's being personalized, that message of personalized medicine, taking responsibility for your own lifestyle and the pet's lifestyle. And personalized medicine is happening in huge ways in the human industry. Yeah, it's been humanizing of pets. You know, when I hear that, you know, advertising executives saying that the food industry is the future of pet, where one sees a family almost cooking for their pet. So we're tailoring drugs to be personalized for humans, and the same thing, the precision medicine, that word of personalized precision medicine, is happening in the veterinary industry. So you need to start taking into account the breed differences. You're personalizing the medicine based on the breed or the breeds of that dog that enters the clinic. So you have a Labrador profile, or you may have a Kelpie cattle dog profile specific for that animal, dependent on its breeds, its age, its weight, its sex. We're doing the same thing with humans with the Ancestry. I'm going to use a lot of quotes because you know, there's nothing like a quote, especially from people in your industry. As Dr. Bell from the small animal practice at Tufts Uni says, genetic testing is indispensable in the vet and encompasses much more than just DNA tests. By understanding and identifying the genetic predisposition to a disease, it will give you as a veterinarian to improve the life of that patient, you know, what it's predisposed to. And it's changed enormously in the last 10 years. And I'll show some examples where, you know, there may have only been a handful of genetic tests that you could screen for. Now there's over 200 that we're able to screen for and predict those occurrences. And by understanding the genetic predisposition, you're able to intervene and act earlier, which is important for your patient. The AVMA passed a resolution in February of 2017. Under the AVMA policy, the Inherited Disorders and Responsible Breeding of Companion Animals clearly states that to maximise the health and welfare of companion animals, it supports the research into genetic and inherited disorders. Important. And to continue the education in this emerging area, it encourages veterinarians to educate breeders, companion animal owners, and the public. Hence this webinar. And as Dr. Hauser has stated, screening pets that are at a risk for developing breed-related diseases allows you as the vet to partner with your client and to develop a personalised life plan based on the genetic profile of that animal. And that's important, that every crossbreed will have a specific genetic profile. Yes, a purebred will have a similar genetic profile, but will vary based on sex and weight and age. But a Labrador will have the same screening, but a mixed breed will have its own unique, its personalised genetic life plan. We need to be reactive in our medicine, and there's a shift happening. It's happening worldwide in the human area. We're going from being reactive to being predictive. And that's the message we're trying to get across here, by understanding the genetic variability, vulnerabilities to disease, you know, whether it's a predisposition to a cancer, you're able to then use that information to help manage your client and in the end, expected longevity of that lifestyle. And, you know, we've gone from fairly invasive to non-invasive means where, you know, a simple blood test can now screen for whether you're carrying a particular disease. So we're moving from a curing to a prevention. That's the message, reactive medicine to predictive medicine. I like to call that forward-looking medicine. And we know facts, pets are living longer. Pet owners have been more proactive. You know, although there's more reading, they're paying more for their animals. Vets are getting better at diagnosis. We know that. Breeders are becoming more responsible. They're more testing. And the key factor for me is that labs are even getting better at providing the services and more tests. And if you look at 2016, the AVMA President Robert Johnson clearly stated that 72% of pet owners viewed the role of a vet as treating their pets when they were sick, and 71% of those viewed the role of their vets keeping them healthy, so they have an active role. Vets play an active role in maintaining that health, being proactive. You know, the age of pets is now increasing. We're seeing that, you know, a Japan study showed that, you know, dogs had gone from living from, you know, an average of 5.1 years to 8.6. And this reflects in recent times where, you know, even cats are now living 2 to 3 times longer than they were previously, all due to better medicine, better diagnosis. One of the things that sort of does annoy me is this thing that we call I refer to it as the Hilton effect, where the humanization of pets. And we're seeing it, whether it's a good thing or a bad thing, it's happening. People are starting to, you know, treat their pets more of as part of their family. Pet parents, you know, from sharing the beds to the way they go about naming. The humanisation of pets is actually an area of great investment. There's lots of money being put into the ability to sell products to those pet parents. But this humanisation does have an impact, and the impact is that it means that people are taking more and more steps to maintain their pet's health. That means visiting the vet more regularly, keeping up to date on vaccinations. Some even invest in special diets, treats, all the grooming factors. It has a benefit in that we're starting to see more uptake. You know, I even get people that contact me that I might see somewhere or see at a park, and they haven't even named their pet yet because they still need about a couple of weeks to decide what name to give it, whether it's Franklin or Ted. That whole personalisation and that humanisation of their pet is interesting. So I'm hoping that, you know, by all this I've passed on. You can embrace genetics and take it on in your clinic and use it as a way to personalize that experience for your client. So I hear this, that it's expensive or costly to buy in and that clients are reluctant to spend on vet care. I don't think that is correct. And let me show you some examples. Here's a group in Australia that have spent close to $215,000 on their French Bulldog. French Bulldogs in Australia have become very popular. Based on that, you can pay up to close to anything between $3,000 to $15,000, such as this one I've shown you in the picture, and was close to $10,000. They see it as a member of the family if I pay that much. So Jonesy had a lung and esophagus problem, was taken to the vet, chronic, required surgery. He was on a ventilator for 3 more times a day. The medical costs topped more than $200,000. It was all covered by a pet insurance company, but they came up with about $10,000 out of their own pocket. So yeah, they were taking 9 pills a day, including antibiotics, allergy. They wanted to see him get better. So these are the pet parents. This is what they're paying. So for me, it all started with these 2 guys who, when I first started, sort of looked like this. And this is, of course, Watson and Crick, the famous people who gave us what we are able to use today, which is DNA. So I'm going to talk a bit about DNA and how it's changed, because that's allowed us then to make a lot of this testing more accessible and available. So I've got a graph here to show you just how DNA testing or human genome sequencing has come down in price over the last sort of 20 years, where pretty much in the late 90s it was close to $1 million to sequence a genome, and then in the space of 15 years, how it's become close to $1,000 to sequence a genome. You can actually sequence your human genome for $1,000 US. It's all been driven by technology, mainly bioinformatics, where statistical data versus the computer science versus algorithms. So with that, we can see we're starting to accept technology. Just amazing, the cost and the technology that's come about. In fact, there's the next slide I refer to is one of my best, because it just shows you there's a TED presentation where it basically goes about that in the not too distant future, maybe about 5 to 10 years, it'll be more expensive to flush a toilet than it will be to sequence a genome. In fact, you'll sit on a toilet and from there it'll take the analysis of your DNA and pretty much sequence that for you. So I think in about 10 years time, what you might see me doing is selling toilets rather than genetics, but we'll see whether it gets to that stage. And it's all been really due just to the advances in technology. When we first did science, we ran what we call agarose gels. We had fairly, insensitive assays where we looked for 1 product versus 2. We then moved to technology, which was all sort of capillary laser based, which allowed us to analyze things in a sort of, in a plexed way and quicker. And today we've moved to technology that allows us to run 1 chip with over 300 tests on that chip all at once. So we've moved to technology that allows us to run multiple tests, analyze hundreds of SNPs, thousands of SNPs on 1 run. So for us, the key material, I'm just going to take you through some basic genetics, is a DNA. It's the root of where all our genetic testing comes from. You can get it from a number of sources. Just a fairly basic slide to show you that you can get it from anything from tissue down to fecal pellets. But what we need to understand is as you move down those materials, the quality and the quantity decreases. So you can't sequence a sample using a fecal pellet. You will need blood or tissue. So in our genetic testing and in most laboratories worldwide, the common sample required is minimal is a swab sample, or you'll see some brushes or cotton swab, and that's sufficient to provide you with enough material to run anything from a single genetic test up to the complex breed ID test, which uses thousands of SNPs. It just captures the epithelial cells off the cheek, it brushes them off, and we extract the DNA off that. The other common material, and we sort of recommend when you're at a veterinarian, is a blood sample. And the blood sample is just a touch better because you don't have any samples that may fail. So be aware when you're collecting a swab sample that you may have a sample where the quality of the DNA is not sufficient because you may have issues such as something in the animal's mouth. There may be some bacterial contamination from exposure to much moisture. Bacteria loves a host, and the perfect host is DNA. It degrades the DNA. So sometimes you may get a sample that comes back and fails for a swab. Hence, we prefer blood collected in an EDTA so the sample doesn't coagulate. So DNA we sort of break up the testing into two forms. So we get the DNA and we interrogate it, and we can interrogate it into what I call practical testing and social testing. The practical testing is when you're actually testing for, parentage verification, where you've got a sire and a dam, where you're looking at disease or risk diagnosis. Have a practical means, and then you've got more of your social tests, which some people might argue the ancestry isn't just social because it then allows you to use it for health related diseases and traits. So they're the two main areas that I see requirements of DNA testing. So DNA testing and the tools that we're able to use are basically very easy, and I'm going to show you just how easy it can be. This is just something taken off the Orivet website, and by using this whole chip, you're able to select what we call a full breed profile. So this is when you have a known breed, you select what breed it is, and then based on requesting a Labrador full breed profile, which is a multiple screen, it's one sample, it's usually a cap price. You would click the button and you would get all these tests that are relevant to the Labrador. So these are all the tests that are relevant and offered for the Labrador Retriever. Very easy, no need to worry about what you're screening for. It does it all specific for the breed. I like to call that the hamburger with the lot, where you're getting it. And then this is just a list of all the tests that are offered, and we call that an attribute profile. So if you have a mixed breed or an unknown breed, we would run all these tests. Very simple, one sample, one swab or one blood sample would run all those. No need to worry about which test am I selecting. You simply nominate the breed or a mixed breed or unknown breed, and all these are run. So let's see where I like people to start confused. So where do you start with this testing? So I might start with the next poll question, and I'm going to show you that 1989 was an important year for us. And not only did it give us the Simpsons, not only did it see the Berlin Wall come down, not only did it see hepatitis C virus extracted, but it was the first genetic test that was identified and published. So for the Cairn Terrier people of the world, it was a test called Factor IX deficiency or haemophilia B, and we identified a variant, which you can see here, the G in red changes to an A. And just so we can all stay, and just to show you that even the science industry is politically correct, we no longer call it a mutation, we call it a variant. We don't like the word mutation, so we say variant. It's detecting the variant. So here's a variant when it changes at that genetic sequence from a G to an A. It changes, of course, the RNA, changes the protein. It then shows you this disease. You're affected for it. So quickly, just some basic science. So again, here's the sequence we target. It's called a substitution. And here are the three types of variants we see. It's going to give you some basic science. You see These are the three types of ways we can assess whether a disease is present or not. A substitution, where, like it was in the Cairn Terrier disease, a G changed to an A, it substitutes that base. You also can have a disease which is a deletion, so the base is deleted. And the other one is when a base is inserted. All those three cause that amino acid protein to change, hence the disease is shown. So that's, I think, important for one of your questions, is the three types of SNPs that we see. So remember, in some of them, you can see more than one base substitution. You may see a huge chunk of DNA deleted. So as I've quickly shown you, it's been huge. The exponential growth in disease testing is amazing. Ostrander said that we should see about 10 diseases annually in 2000. I can tell you it's almost close to 20 to 30 new diseases seen annually. So currently we sit, Orivet, at about 178 diseases in dogs, with another 39 to be added the next 3 weeks, just to show you how quickly they're growing. The exponential growth is scary. And like always, the dogs seem to win the race against the cats for some reason. Just a lot more clean sequence available. You can see close to 222 tests currently available, and all in cats about 100 available. They just keep on coming. Just a quick couple of new ones that you may have all want to know about. JME and the Ridgeback. I'm going to see if you can see this video, which is just a myoclonic epilepsy. You can see this this Ridgeback, which is showing some little fits there, collapsing. So again, a test you can now test for, available. It's been available for over 12 months. And then there's also the NAD in papillons, which is an ataxia, and you can see starts to show some tremors. Again, it's trying to walk about in its gait, and again, we can test for genetically. Hopefully everyone could see that. So most of the confusion, I know, is what's available, what's published, what's patented, what's breed specific, who offers the, which is generally the poll that I conducted. Most of you would be about these questions. So let me start. Our biggest problem is that most people go to the internet and they diagnose and assess what they do off the internet. It's not a bad start, but let's see that you can't go past a good book. There are 3 books that I recommend and I've used, and these are 2. There's a dog one, a cat one, and there's an Orivet one that we've done, which is a really nice quick reference guide available to vets. If anyone's in this webinar would like a copy, I'm happy to send them out one of the Orivet ones at no cost to them. Excellent quick reference guide. Most vets that have seen it love it because it's basically a 1 page on each of the common genetic disorders. Very easy to use. You can't go past some publications, and I'm going to quickly just go through. There's a nice publication because I'm sort of going to be close to time, which shows you the Ancestry of Dogs, where basically they've broken up, you know, breeds, 160 odd breeds, into what we call 23 clusters or groups. And it's not surprising that the clusters all fall within the way the geographical locations and the physical characteristics of these breeds. So if you're to look at what is the most ancient breed, we start down here, where the Basenji looks to be the most ancient breed, and then from there we evolved to some of the Spitz breeds, the Arctic breeds, the Asian breeds. So we've got the Tibetan Mastiffs, we've got the Siberian Husky, the Akita, the Chow Chow, and then from there we started to move to some of the more lap dogs that you can see started to produce, and then we move around. So you can see the bull breeds. This is an excellent little chart to give you an overview of just the development of breeds. And, you know, with that, it's important to understand that with that comes certain diseases. So, you know, collie eye anomaly is associated with most of the herding breeds, which you can see here. Excellent little chart to sort of give you an overview of breeds and their makeup, and with that comes the disease association. So where do you start? I'm going to give you a couple of good sites. Use these websites to access some excellent information. you've probably all gone to this website and looked at it. You can then select the diseases. You can look by breed. There's an excellent website at Penn Gen, which pretty much updates all the information every 3 months. You're able to select a breed. Then based on that breed, you can then look at the diseases that are published and specific to that breed. You then click on that publication and view what you want. Excellent little tool. You should all bookmark those 2 links I've given you. So if you have a Labrador that comes in and you want to know about a particular disease, or we're going to see what diseases are relevant, excuse me, they're all listed here. What is published is available. There's also an excellent local website, which is fantastic, developed by the University of Sydney here in Australia, the OMIA website. You click on the number of disorders by dog, it then shows you what's available genetically, it gives you publications. Excellent, 3 excellent websites that you should all bookmark and use to make it less confusing. So as I said, you can click on any of those. You click on any of those diseases, as you can see here, it lists all the, in alphabetical order, from cerebellar ataxias to, you know, cone degenerations or achromatopsia. You then click on that link, and then it gives you all the information. It gives you whether it's recessive, whether the variant's been published, some molecular basis, the pathology behind it, an excellent website. So we're all starting to feel good about scientific publications, and we all love the work that scientists does, and you know, these people are the gatekeepers of genetic information. We all believe that everything they do and everything is published is correct. There are some ways of validating those, but I'm going to show you sometimes where something can happen, and it happens in publications. For example, you're all aware of the MDR1 ivermectin genetic test that's available. You've probably all used it. Well, it's interesting because there was a publication in 2003 that was published in the Journal of Internal Vet Medicine, which had the correct variant. And then there was another publication in 2005, which unfortunately had the incorrect variant. So you can see instead of an AGAT, they had typed an ATAG. So if I was developing my test on the 2005 publication, I'd be offering the incorrect test. And I'm going to show you some things that you should look for when you look to see whether a test. So typos, they happen. They can happen in, as shown in this newspaper article. You know, we need to make sure that the publications are validated. But sometimes there can be typos. Sometimes the labs need to report and make sure they're reporting correctly. And I'll show you what we do. There's some other tests which are published where the test published is benign. There is no association of that variant to the disease. You know, we see a staggering 12% of miniature schnauzers test as positive for this type A PDC variant, but the disease is benign. You do not see any of that associated with, again, published. So again, it's the way laboratories report this. There's also some publications that have been retracted based on the study design not employed, the validity of control samples. So again, we only work from offering tests that are published, but again, some of these may have issues, and we hope that most of these are retracted. So I've got about 5 minutes. I want to try and hurry up because there's important things. I'm going to talk about another bit that confuses people, which is interpreting the results. And it is confusing because one place says mutation, one can say variation, and in the end it's all confusion. Because, for example, here's a way I can report a positive result. You can call it positive, affected, at risk. I've seen plus, I've seen mutant, I've seen people use abbreviation. It can be confusing. So laboratories need to think about standardizing. For example, this is the way I've tried to find something that's similar to genetic testing and interpreting those results. So we as a lab get a result and it comes back as what I call the raw data. So I've shown you how we interpret whether it's a G or an A or a T or a C or a deletion or insertion. So here's 4 genetic results we've received. The lab is then asked to report that into some sort of salad, which is this interpretation. So all these results are about a particular color interpretation, and then the owner wants us to not only interpret the salad, but put the dressing on for them and tell them whether it's a seal mink tabby carrying cinnamon. Yet the lab is only responsible for this data that we call the raw information. This other information is something that the client wants and something that the vet shouldn't be concerned about. This is what they need to know. So how do we make it easier? You need to look at the report. So this is where we can help. So here, for example, is a cat that's tested for hypertrophic cardiomyopathy. The result is affected or at risk 1 copy, and it's a dominant disease. It tells you what's been detected. It tells you the publication and what variant you've detected. So if I was reporting an incorrect publication, you could compare it to another laboratory. It gives you some information about that disease. It gives you the background, and then it gives you the recommended breeding. That's sort of the standard you should look for in a report. So the information that a veterinarian has is reliable, accurate. It gives them the information they can then pass on to the pet owner or the breeder. We also take a lot of, we have a process that we have an approved collection method. Just quickly, if an owner submits a sample themselves, it's not an approved collection. Sample integrity is important for us. If a vet collects it with the microchip, it has an approved collection method. Positive ID, independent collection. There's no use comparing a result that's been collected by someone themselves without a microchip to an animal that is collected by a veterinarian. We have an explanation of all the results. We clarify the genetic testing. We spend a good 2 or 3 pages on it. Most clients don't read past the first page, which is the result. We clarify all the key messages, such as genetic heterozygosity. We also hear that some vets say it's not relevant. Why should I do testing? Well, I'm just going to let you into a little code of practice that's here in Victoria and most of Australia, where it's an offence to sell an animal with a hereditary defect to a person without that person being aware of that defect. So it does have a role. It's an offence to sell an animal with a hereditary defect to a person without the person being aware of that. So you can breed an animal that might be positive for VWD, but the person purchasing that animal must be aware of it. And the code has the veterinary practitioner involved. I'm going to try and hurry up, Hannah, because I think I've only got 5 minutes. Is that right?
I mean, you can take your time. We have another, I would say, 5 minutes. Yes.
Okay, good. Well, I will. So again, most vets say not relevant. They don't see the disease. Again, I'm going to show you some statistics. Degenerative myelopathy, which was firstly seen and very common in the German Shepherd, can now be screening close to 80 breeds. You know, we have a carrier and at-risk percentage in the dogs that we've screened at close to 40%. So again, I hear a lot of not relevant. It is relevant. There are dogs that show the disease. Again, EIC, we have about a 31%, about a 31% carrier and positive at risk for that disease. And people say not relevant. Well, a lot of retrievers are used and for working purposes. And retrieving, I've heard cases of where, based on them going out and over exercise, you know, dogs collapsed and drowned while retrieving. So it does have relevance. You can. These are only 2 that I've selected. And the way screening can assist, think of it this way. So you get an animal that comes in with a bleeding or clotting disorder. You can simply screen for an attributes profile and get and we break down the diseases into the disorder. Here's a list of just some of the genetic ones that are relevant. You can get all those done, which are relevant to over 70 breeds. So even if you have a mixed breed and there's some standard poodle in there, or toy poodle, or West Hound or White Terrier, you will have the ability to at least eliminate these diseases via a genetic test. So again, you can break them down in disorders with over 160 tests. There are some disorders that have close to 40 genetic screens for them. And I hear some vets sometimes saying, oh, the genetic tests are not clinically relevant. Well, here's a publication of pedigree versus non-pedigree, of what the veterinarian sees and what the owner can, and things like congenital disorders, welfare, ocular diseases, blood disorders. I've just shown you some musculoskeletal disorders, nervous system disorders. So you can see some of these that are offered, so that have been ticked or seen as being seen in breeds by owners and vets. We offer screens for. There are genetic screens for them. So here's an example of some tests that are available, and I've shown you basically all the tests that are available for musculoskeletal. So you can see, this is what's available in the pedigree and non-pedigree, and again, an example of just the musculoskeletal diseases that are available via genetic tests. So I'm just trying to paint a picture. They are, it's easy. You don't have to request, one particular test. You can screen them all based on the current technology. And most of this technology is available through some of the organizations we do, and there's probably a few others in the US that offer these tests as well. So people always tell me that, there's limitations. The DNA tests are based on preliminary or assumed relationships. What is the thing? You get an animal that's at risk. Does it mean it will develop that disease? No, it doesn't. It means it's at risk. It means a veterinarian needs to assess to see whether those symptoms are being shown. And as we clarify in our genetic testing, what causes some of those to be shown at 3 months, what causes some of those to be shown at 3 years, and some you may never see those symptoms, is a combination of genetics, environment, and even nutrition. What switches that gene off and on, we just don't know yet. So for example, the German Shepherd, as published in Pharrell, has the largest number of disorders, and in blue I highlight what is available via a genetic test. So close to over 12 of those are available via a genetic test. If you look at the Australian Shepherd, almost 80% of the disorders identified are available via a genetic screen. So there's relevance there. It's relevant. You can screen for these via a genetic test, and they're common to that breed. So I've highlighted the 4 common breeds, and you can see from the way research is directed that pretty much most organisations will research and develop a disease for a breed that is fairly popular. There was also a publication through where people go, oh, it's not relevant, you don't see me mixed breeds. Well, through a publication in this journal in 2016 and screening mixed breed dogs, we were able to identify that these diseases were present in breeds that weren't published. So for example, CMO, which is available via a genetic test, was identified in the publication as only being in the original scientific publication as only being seen in the Cairn Terrier, Scottish Terrier, and West Highland Terrier. And through screening of mixed breed dogs, they identified them in a Lancashire Terrier and an Australian Shepherd. So now those 2 breeds are included in the screen. Factor 7 was originally only published in these 5 breeds, and now is seen in another 10 breeds. And in the Finnish Hound, we saw 54 out of the 237 screen was shown to be positive for factor 7. So there is relevance in some of those mixed breeds. And one of the most common, what I call developmental breeding, is the Labradoodle. Here's Mr. Wally Conran, the person who developed the Labradoodle through And some of the new ones we tend to see are now the Gerbian Shepsky, which I've been told about is a German Shepherd cross Siberian Husky. And when I asked the person about them, he said they're an attractive guard dog. Interesting. It's a nice way of describing a Gerbian Shepsky. So I'm going to finish up with just a few little things, and I'm going to show you that, you know, we continue to develop new diseases. Science is evolving. You know, you can't really stop scientists from doing what they want to do. I've probably shown you some good areas to source some of this scientific information. I don't want to promote Orivet, but I will. Go to our website. They're all listed. They're all listed by breed. We give you the publication. We list the variant. But since my talk was had a title about a pet swallowing genetics, I thought I'd better try and find something that's relevant to my talk. And here is a dog called Macey. And Macey, in February of 2017, the 3rd, swallowed an 8-inch knife and lived to tell the tale. So apparently the dog owner said that they must have left something on the knife, some food, and was put in the dishwasher. And like any good little puppy that's hungry, it smelt the old food on the knife and swallowed the whole knife. So, and as the vet said, they've never seen an X-ray like Macey's. Extremely lucky to survive. And the reason it survived, according to the vet, is it swallowed the handle first. Interesting. So that's a good little way to finish the talk. But I thought before I go, I want to sort of talk a bit about breed ID. I don't know how many vets have used a genetic test to identify breed ID, and some will argue that, you know, we don't need it. Sometimes you might get a result back that sort of doesn't fit with what you think is right. But I'm going to show you an example of where, you know, if you look at the studies, guessing the breed is actually can be quite poor. And in fact, studies have shown that people that in pounds who identified breeds based on physical appearance tend to get about 80 to 90% incorrect based on the DNA science. For example, here is a dog called and here is a dog called Biggins and a dog called Dewar. So Dewar was purchased as a Staffordshire Bull Terrier and looks nothing like a Staffordshire Bull Terrier and was actually a Kelpie Heeler cross. So you've got to understand, this person purchased it as a Staffordshire, doesn't suit their environment. A lot of the time, dogs that end in pounds because it's doesn't suit their lifestyle. So this dog was sold as a Staffordshire, and based on that photo, I could tell it wasn't, but it turned out to be a Kelpie Heeler. And then we have Biggins was sold as a Hungarian Vizsla. So tell me that dog was like a Hungarian Vizsla. So you've all probably heard about the DISC personality types, and it's been interesting how we at Orivet are able to break down our clients based on their breed into these types. So we find that the Labrador people tend to be the more influential, in that they're happy to collect a sample, go through a vet, clarify results. We find the Labradoodle tend to be the most sort of conscientious as well. We have the dominant type, which is the cattle dog people. And then we have the newer breed, which is the French Bulldog, who demand and want results as of yesterday. So again, I just want to finish on just some client stuff. Hannah, I think I'm finished there. I'm happy to leave it at that. I'm happy for people to email me any questions. If there are any veterinarians that want to ask any questions now, I'm happy to maybe take 2 or 3 minutes. If they want to email me for that little gift I promised during my webinar, I'm happy to send them out one of the Orivet books.